So I'd like to begin today with a land acknowledgement. Rensselaer Polytechnic Institute resides upon the ancestral homelands of the Mohican people, who are indigenous peoples of the lands of New York. Despite tremendous hardships and being forced from their lands, today their community resides in Wisconsin and is known as the Stockbridge-Muncie community. We honor this community, past and present, and are committed to building a more inclusive and equitable space for all. So the BioArt Talks at CBIS are a series of online presentations given by international recognized bioartists. Bioartists work with biological materials and use biotechnology to produce their artwork, looking at the materiality of biosystems and the processes used in biotech and also in scientific research. So I'm a longtime bioartist, so this is why I'm interested in having all these wonderful people come and speak with us, because selfishly, I want to hear what they're up to. The BioArt Talks artists have included Orkan Han, Heidi Boisvert, Aysen Karo Chashin, and today's guest, Anna Lindemann, and upcoming next week for our final one will be Mary Magic. This series has been generously supported by funds from the New York State Council on the Arts by approval of the New York State Legislature and the governor, and we are very grateful for this support. So let me introduce a little bit about Anna Lindemann. So Anna, who's here with us today, calls herself an Evo Devo artist. Her work as an animator, composer, and performer explores the emerging field of evolutionary developmental biology, or Evo Devo. Anna graduated magna cum laude with honors from Yale with a BS in biology before receiving an MFA in the integrated electronic arts from Rensselaer Polytechnic Institute. Yes, she is one of our own. And we're very happy to have her back. She is currently an assistant professor in the digital media and design department at the University of Connecticut, where she has pioneered courses integrating art and science. Anna has presented her Evo Devo art at such venues as the Imagine Science Film Festival in New York, the ICEA Conference in Vancouver, KLI Institute for Theoretical Biology in Klosterneuburg in Austria, Evo Muse Art in Parma, Italy, the Experimental Media and Performing Arts Center here at RPI in Troy, S Cinema International Film Festival in Australia, the State Darwin Museum in Moscow, the Franke Program in the Science and Humanities at Yale University, and finally the Istanbul Fringe Festival, among many others. She is the recipient of a Connecticut Arts Fellowship, and her animation work includes commissions from the USS Constitution Museum and Boston Cyber Arts. So a little about today's talk, Evo Devo Art. Anna's work seeks to uncover narratives with rigorous scientific research, to visualize biological processes in novel ways, to define new artistic creative processes modeled on biological processes, and to examine the human emotion and subjectivity behind scientific research. So as part of her talk, Anna will highlight several Evo Devo artworks, including The Colony, an art science performance about sisterhood and the evolution of social life in ants and humans. I'm so looking forward to this talk. So please join me in welcoming Anna Lindemann. Woo!
Thanks so much, Kathy. So let me see if I can get set up with all the screen sharing. Give me 1 second.
That looks great.
Right, hopefully people can see things.
Yes.
So I'm so excited to be part of this talk, and thank you so much, Kathy, for curating this series of talks on bioart, and also for all the entities that made this possible, the CBIS and the Sanctuary and the Nature Lab, and I'm grateful for this support. So as Kathy said, I'm going to be talking about Evo Devo Art, and this is really my own take on BioArt. So it's art that is inspired by and modeled on the biological processes of evolution and development, known as Evo Devo. So this is a relatively new scientific field, about 20 to 30 years old. And Evo Devo itself exists at a kind of cross-section. It draws upon embryology, evolutionary biology, taxonomy, computational biology, paleontology, to name a few of the disciplines that kind of come together to study these two interlocking processes, the process of evolution, which is acting at very large timescales, so hundreds of thousands of years or millions of years as species get defined, and then the process of development, which is over the course of an individual lifetime, acting just over the span of a lifetime. And evolution itself can act on development. So this is a complex puzzle, and it's one that I like to explore in my art. And as Kathy mentioned, I take a kind of transmedia approach. I work as a storyteller, animator, composer, and performer to explore Evo Devo themes. So today I really want to highlight 3 projects very briefly to give you a sense of the range of my approach, and then I'll discuss one project in greater depth, which is the colony. And then I'm mostly excited for you to ask questions, so please do chime in with questions, and I'm looking forward to hearing those from you. So to start, my work creating Evo Devo art really began when I was a student at RPI, studying with Kathy and Michael Sentry here and others. So it feels really especially great to be back amidst the RPI community for this series. And I created my art science performance Theory of Flight while at RPI. And this performance explores the development of wings and the evolution of flight in birds. And it also examines a kind of human emotion and subjectivity behind this scientific research. So the performance begins as a lecture on wing growth, but the lecture goes awry pretty quickly as the scientist character I play reveals her own personal investment in flight, and we can see where that takes her with her wings here. So I'm going to play just the start of a preview of this performance so you can get a sense of kind of the aesthetic.
In 1948, Saunders was conducting experiments on chicks, and he found special cells in the side of the developing chick that seemed to define where a limb would grow outward. And if you move those cells to a new location, say the back of the developing chick, you get a new wing growing in that new location. TVX5, FGF10 can give rise to a whole new wing. I think it has all the properties that I need. DBX5, FGF10, DBX5, FGF10, DBX5, FGF10, wing gene network plasmids. DBX5, FGF10, DBX5, FGF10.
I forgot to check, did the audio sound okay? It seemed like I did the share screen. All right.
Sounds great, thanks.
Thanks for the confirmation. This next project is called Beetle Bluffs, and it's an animated short that I shot at the Museum of Comparative Zoology at Harvard. And the animation brings to life research on beetles conducted in the 1930s by a former curator of the collection, Philip Darlington, who used to sit at this very desk here that's hidden in the back of the collections. And the animation explores really what I think of as an amazing evolutionary story of 9 different beetle species from very different parts of the evolutionary tree that converged on a color pattern that helps the beetles avoid being eaten by a lizard predator. So the music for this animated short uses a generative approach to music composition inspired by gene networks, and I'll talk more about that gene network music at the end of the presentation. But for now, I'd like to play a short 30-second clip from this 12-minute short, and you can see the evolutionary mimicry in action. The project I'm currently working on is an animation with music about food webs in the Arctic and how the food webs might change as the Arctic environment changes. And this is a project I am developing in collaboration with Dr. Alice Duvivier, who's a polar scientist and a climate modeler at NCAR, which is the National Center for Atmospheric Research. So these are just a couple of style frames from this project that's in process and ultimately will become an animation. So the Arctic regions are the places on our Earth that are changing and warming the most rapidly of our entire planet. And this is as a result of human activity. So this change in the Arctic is resulting in shifts in sea ice coverage. There's less sea ice in the polar regions, and also changes to the food webs. So our animation is going to explore the Arctic changes at different scales, from this global scale seen here to the melting of individual ice flows, to the microscopic scale, focusing specifically on microscopic organisms at the base of the entire food web. So phytoplankton, like these jolly centric diatoms. And the animation is also going to explore speculative changes to the ecosystem. So changes that aren't really incorporated in scientific models yet, but are based on scientific research. So one example is phytoplankton species that are found closer to the equator becoming more prominent in the Arctic and overtaking Arctic phytoplankton species. So here we have some coccolithophores, which is a word I just love saying, dominating the diatom population. So looking at these three examples, one might ask, why make Evo Devo art? And I think there are many reasons, but I'm going to list 3 reasons that motivate me and that I think are important. The first reason is that I think this is a form to share amazing biological stories with wide audiences, to get lots of different people excited about these biological stories. The second reason is that I think this is a means to harness biological creativity for artistic creativity. And by that I mean that if I turn to processes like evolution and development, I might expand my own creative possibilities. So Darwin famously said that through evolution, you get endless forms, most beautiful and most wonderful, occurring in nature. So if we're able to harness this evolutionary process for art making, perhaps I have a chance to create artistic forms that are endlessly beautiful and wonderful. At least that is the hope. That's the aspiration. And then the third reason is that Evo Devo art provides a powerful means to connect the human experience to other biological systems, to reflect in a way that perhaps scientific publications can't necessarily reflect on the link between our own experience and that of other creatures. To dive into this a little bit more fully, I'd like to share a closer look at one project in particular, and that is The Colony. And this is a show about sisterhood and the evolution of communication in both ants and humans, which are two of the most social creatures on Earth. And this performance fully embraces an Evo Devo Art mentality. Here I am at a moment in the show that is a kind of animated fable on the evolution of complex social life in the ant colony and the emergence of castes. You can see a queen, a worker, and a soldier there being highlighted. In this performance, I play the role of Mona, who is a character who is trying to reconnect with her estranged sister, and she turns to the ant colony for inspiration. So she thinks that surely an ant colony that has 500,000 sister ants that swarm together in search of food, they migrate together, and they even reproduce as one big superorganism, that surely this biological system can help her reconnect with her sister. So I'm going to play the preview for the performance. It's about an hour-long piece, and then the preview is a few minutes.
I should have done this months ago, written to her and that sticky little toddler of hers. I'm the worst aunt.
Did you want to know more about your personal failures?
Definitely not.
Did you want information on ant communication?
We ants by bulk, by thousands, many mandibles, none to speak. Our eager eyes are eyeing, but our vision's poor too weak. Wordless we work together, a swarming plural one. We nomads of the jungle, deaf to bird song, blind to sun. Their unseen trails we're trailing, pheromones rout our rendezvous. Our chemical cartography, our sign and signal too, our tongueless mother language, our prime directive, our call to fight, from glandular epitheliums of abdominal sternites. Each ant supplants another. One goes forward, one reverse. Roots and leaves, beware of masses. There's no ground we won't traverse. We ants, a horde, a chorus. We glad colloquium. We insects, orders, we sisters can dance all together, all as one. We ants, a horde, a chorus, all together, all as one. We ants, a horde, a chorus, all together, all as one. We ants, a horde, a chorus, all together, all as one.
So I'd like to share a bit about the process of creating the show, including some of the inspirations and some of the details about developing the narrative and the music. So this performance was inspired by the Carl W. and Marion E. Rettenmeyer Army Ant Guest Collection. So this is a mouthful, but a really amazing natural history collection amassed over 50 years of field research in Central and South America by the Rettenmeyers. And on the right here, you can see a photo of Carl and Marion Rettenmeyer, who were scientists and partners in life and work, and devoted themselves to studying army ants and their associated species known as guests. In the center you can see some of the components of the natural history collection. So that includes pinned specimens, specimens in vials, microscope slides, and Kodachrome slides, so photographic images, as well as very extensive field notes on these field cards. Over the course of their research, they identified 557 animal species that live on and with a single species of army ant, Eceton burkellii. And this is the largest known animal association centered around one species. So living along with Eceton burkellii are many species of mites, as well as beetles and birds and butterflies, all kinds of species that rely on the ants for their survival. So this is a complex biological system, but also an under-recognized one that is worthy of celebration and further investigation. In 2015, this natural history collection was donated to the University of Connecticut, where I'm an assistant professor in the Department of Ecology, Department of Digital Media and Design. The Department of Ecology and Evolutionary Biology worked to preserve this collection and received a big NSF grant to secure the future of the collection. That meant putting all these vials that had evaporating liquid in them into proper archival vials and digitizing the database as well. So growing out of this donation was also something called the Ant-U Initiative, where many people from across the university came together to create projects inspired by the collection. And from 2017 to 2020, I served as a co-director of this Ant-U Initiative, and we created 16 interdisciplinary projects, one of them being the colony. So when I began developing the colony, I really started my own research by investigating the Kodachrome slides. There were 6,200 Kodachrome slides in their collection, and I started by just looking closely at them. Here is one of the images I came across that was especially striking to me, this image on the left. This is an army ant queen, and you can see that her abdomen here has swelled to accommodate somewhere between 20,000 to 200,000 eggs. And she's surrounded by these papery pupa, and then also being tended by these little worker ants and soldier ants who are her sisters. So except for a few weeks a year when a handful of males make an appearance within the colony, the rest of the ant colony is entirely female. So about 500,000 ants that are the queen, the workers, and the soldiers are all female. So this image immediately triggers all kinds of reflections, complex reflections about sisterhood, about motherhood, about the balance of work and childbearing, and about hierarchy that exists within social life. And these reflections about the ant colony also informed the kind of human story within the colony and reflections on motherhood and sisterhood within human societies. In the performance, this particular image inspired an aria from the perspective of the ant queen. So thinking about what would it be like to be this queen who has an abdomen swell so much that she can't even really move and is giving birth to a whole new generation for the colony. What would that experience be like? And in the context of the colony, we see a projection used to transform the performer who plays the role of my sister into a kind of ant-human chimera. And so I'm going to play a short excerpt from the Ant Queen aria.
Contort, expand, exert control. I swell, I grow, it
So ultimately, the narrative for the colony is an Evo Devo narrative that draws from the Rettenmeier Army Ant Guest Collection and also from a wide range of scientific literature. So this includes articles about army ant behavior, about caste determination in ants, and the evolution of eusociality in both humans and insects. So eusociality being a specific complex form of social life, often characterized by altruistic behavior. And different people have different opinions about whether humans are truly eusocial or not, but a lot of people think they are. I collaborated with writer Emma Komlos-Robsky in the creation of the script for the colony, and here you can see us forming a bunch of our notes and brainstorms about how do we translate these Evo Devo ideas that are in the scientific literature into a narrative that connects to the human experience and to human emotion. So in one scene, we did that process of translating by highlighting the environmental and genetic factors that determine ant caste during development, alongside positioning Mona, who's the character I play, who is usually a solitary person, in the novel role of caretaker. And here she finds herself in charge of a collection of larvae. So based on evidence that the amount of food a larva eats determines the larva's caste as an adult, here Mona attempts to control which larvae will become workers and which larvae will become queens by feeding them different numbers of jelly beans. And of course, we can judge her choice of nutrition, which may be misguided, but that's just the kind of character she is. Based on evidence that in certain ant species, larvae that become pupa later have longer to eat and will be more likely to be queens, here Mona explores the application of hormones to a developing larva that can trigger the onset of pupation. So two hormones throughout insects are really involved in pupation: juvenile hormone and ecdysone. And in the show, Mona uses a kind of watercolor version of these hormones to define the fate of these different larvae. In another scene, a kind of animated fable traces the evolution of complex sociality in ants from two sister ants living together in one nest, here on the left. So the kind of very beginnings of a colony with just two individuals, to the evolution of a division of labor. So a division of labor between foraging, we see an ant bringing back a cricket leg here, and reproduction. We see another ant staying in the nest and laying lots of eggs. Ultimately, this small colony evolved into much larger colonies with hundreds of thousands of ants, and large colonies required also the evolution of some form of communication system. So ants rely on pheromone paths, which are invisible chemical traces, but in the performance, these pheromone paths are represented by pink yarn. And the same pink yarn becomes the kind of communication path that ultimately reconnects Mona and her sister. So the performance also takes inspiration from communication technologies and the evolution of human communication, and it includes a whole range of ways to communicate. So one method is the Flutterdex, which is a kind of mashup of Facebook and Rolodex. There's tin can telephones, old telephones, letter writing, and of course the pheromone paths of the ants. So I'd like now to talk a little bit about how Evo Devo music, or Evo Devo, inspired the music, as well as the narrative for the show. So the music for the colony brings together live performance by a soprano Lucy Fitzgibbon and pianist Ryan McAvee McCullough, and the show was specifically created for these two musicians, which are really exceptional musicians. And they perform alongside synthesized accompaniment created with the gene network music. So we heard a little excerpt of that at the beginning with Beetle Bluffs that was shot in the Museum of Comparative Zoology, and that same process is used in the colony. So to explain what gene network music is, what do I mean by genes? We're going to have a little moment of Bio 101 here to review where this inspiration comes from. So genes are sections of DNA that code for proteins, and DNA is transcribed to create RNA. RNA is translated to create proteins. Different kinds of proteins are produced in different cells, and then these different cells are organized to form a complex organism like a fish or like you or me. But it's not entirely obvious from this sequence of relationships how you get the correct mix of proteins produced in the right place. So how do you get proteins related to an eye forming in the head rather than on the tail of this fish, for example? So to better understand what triggers the production of certain proteins, let's take a look again at our strand of DNA. So we know that RNA is produced from DNA, and then proteins are produced from RNA, and then certain proteins can act as regulators. They can bind to sections of DNA, and that can serve as a trigger to activate another section of DNA. So that DNA being triggered will form a gene expression, or we can say that the protein is expressed. So you get here a self-regulatory process where proteins regulate the production of other proteins. And this regulatory process can be represented in a gene regulatory network, or gene network for short. So here we have a gene network consisting of 7 genes, and this is a relatively simple gene network. Our human genomes have something like 20,000 genes, so 7 is a small number in comparison. And we can see different kinds of interactions represented here. In this example, we can see that this gene 1 or protein 1 is activating gene 2. Gene 2 is activating gene 4. And then we also see these plunger symbols. So gene 3 is trying to prohibit the activation of gene 2 and prohibit the activation of gene 5 here. So what happens when the interactions within this network unfold over time? Well, at any given point in time, some of the genes will be on, they will be active or expressed, and other genes will be turned off or inactive. And we see that what results is that this network cycles through, in this case, 3 different states, where each state is a certain set of genes being turned on. So we get a kind of 3-beat pattern: 1, 2, 3, 1, 2, 3, 1, 2, 3, a kind of waltz. And we see the musical and rhythmic potential for the gene networks here. So for gene network music, I simulate the gene networks in MATLAB, and here you can see a screenshot of some of the code of initializing the gene network. And then these gene networks are run over time. So here we see gene expression over time for a gene network with 170 genes. So on this vertical axis are each of the genes, and we can see, for example, at the top here, this gene has a little moment of color. It's turned on, and then it's blank. It's turned off, and then it turns on, and then it's blank. And we see that unfolding over time. So this is for 170 genes, and we see different color coding here, and that corresponds to some subnetworks. And I can talk more about that, but briefly, that's just the amount of connectivity. So a subnetwork will be more connected than another subnetwork, and the different subnetworks work at different time scales. So we can see that this gray subnetwork updates much more slowly than these subnetworks up here, the blue and the red. And we can see different patterns start to emerge. So here we can see quite clearly a rhythmic pattern, a repetition that is happening between some of these yellow genes. So this gene expression over time can serve as a kind of musical score, a kind of rhythmic score. And we can interpret the score by mapping different of these genes to different pitches or different instruments. And then we can listen to this gene expression over time. So in this instance, we have a score that is 70 beats long and contains 1,784 notes. And some of those notes we might hear, and some of them might just serve as controllers. They might just be regulators. In the context of the colony, I combine gene network music with traditionally composed music. So this is a little excerpt from one movement where at the top you can see the soprano part, and that's traditionally composed. So that's just me picking notes that I think are appropriate. And then at the bottom are the parts for the pianist, as well as for some synthesized parts, which are generated using the gene network music. So the parts for the keyboard player here, each pitch corresponds to the keys on the keyboard. At the bottom here, there is a special note mapping. So as an example here, this synth voices 2 has the following note mapping, where this C corresponds to a pitch that is a B and to a sampled part of speech. We can see here very small we ants, a chorus, colony, a horde. So these fragments of sampled text are mapped onto this kind of custom digital instrument, and then we get what I call an ant chorus, because different fragments of voices are played through the gene network. So I want to play a short excerpt from this movement, and as you hear the gene music, you're also going to see some visuals inspired by the army ant swarm raid, and the visuals directly reference this diagram of a swarm raid that was included in an article by T.C. Schnerle, who was the mentor of Carl Rettmeyer, so the scientist that created the natural history collection that inspired the project. And in this diagram, it's a kind of rare visualization of the invisible paths, the invisible pheromone paths of the ants. And you can see at the beginning here, this is the first stage of the swarm. This in the center, this is a kind of node that represents the army ant's nest site, known as a bivouac. And the ants then migrate out in all directions, searching for food. And then they find a particular path that they want to pursue, and they go along a column and then fan out again to gather food and then bring it back to the nest. So 5 hours in here, the ants are going past along, this is a long fallen tree or something, and then they're fanning out. This is about 55 meters from the bivouac site or the nest, and then they come back to the nest. In the context of the colony, we see the visuals of the ants' pheromone trails here morphing into written text. So creating a kind of connection between the ant communication and then the human forms of communication, in the same way as there's a kind of connection musically made between the gene networks, this biological system, and then also fragmented text. So we see that both in the visuals and in the, and we also hear it. So we'll play just a short clip from this song, Ants Can Fix It.
Ants fix it, fix it, fix it. Ants fix it, fix it, fix it. Ants fix it, fix it, fix it.
So I'd like to end by playing one other clip from the colony that gives a sense of the range of application of the gene network music. So we just heard an instance where there was kind of a vertical layering of traditionally composed music and gene network music. In this instance, there's some gene network music that gets introduced as these swells that kind of come and go. So here's the excerpt from the score, and then on the right here is a screenshot of Logic, the digital audio workstation that I used. And you can see that there are different little swells that are happening in the music as it goes from start to end. In this instance, the gene network music isn't mapped onto sampled voices. Instead, it's mapped onto some percussion, some pizzicato strings, so plucked strings, some bowed strings, marimba, and piano. So a different range of instruments are being mapped onto that gene expression output. And we're going to listen to just this last section of this song. And this is a music and video sequence in which Mona goes to the grocery store in search of more nutritious food for her larvae. And once she arrives at the store, she finds the patrons of the store kind of transforming into swarming ants, bringing offerings of food to their ant queen, who is none other than her sister. And we're going to listen to the last section of this piece. So if this presentation has piqued your interest and you want to watch the whole performance, you're in luck. It's currently streaming through the Gorilla Underground, the 2022 season of the Gorilla Underground, which is this great virtual performance series produced through Gorilla Opera, and it will be streaming through May 31st. And by coming to this talk, you can get an offer code. This pay what you want, go15, so PWYW go15, to pay what you want and watch the whole show. So now I'd love to open it up to questions. I hope this is giving you a taste of my world of Evo Devo art, and I look forward to hearing your questions. And I'll put that link to where you can watch it in the chat as well.
Thank you so much, Anna. That was just so delightful and so informative and so inspiring. I think everybody, there's some wonderful comments in the chat, and everybody has just been really mesmerized from all of your work. Thank you, thank you. Yeah, everybody, please speak up, raise your hands, or add comments or questions in the chat. I'll just start us out because the work is so just fun and really, really wonderful to engagement across the board. I particularly love the colony, but also all the other pieces you showed. So, there's a way in which you capture this kind of exuberance and excitement that scientists have about their own work, like about their own research, that they're really completely passionate about it. And, as artists, we know that feeling too. I'm just curious for you, was there ever a moment in your history where you were thinking, maybe I should become a scientist, but then became an artist? And what was that kind of like little voice in your head about, how did that artist scientist person win out instead of the scientist artist, or whatever. Maybe they're both there. Maybe it doesn't mean anything. Maybe you're doing both.
Yeah, I think you characterized my journey perfectly. I think there definitely was a time where I thought that maybe I would go into, biology research. I studied biology as an undergrad and worked in a lab, and was super fascinated by these stories and continue to be fascinated by them. I think my experience working in the lab was that there were many depressed postdocs who were struggling with their experiments, and I was like, oh gosh, this just has a bleakness to it, where there was the biology is, there's so many factors at work that even if you try and design a really great experiment, things can just not work, and you can be hitting your head up against a wall for so long. And I think it's hard to realize that until you work in a lab, because so much of what we see of the science is that 100th time after the 99 things that didn't work, the 100th time that did work, and that revealed some kind of new insight about a system. So I think I continued to be really excited about the biological systems, but I found that maybe the bench research, at least, wasn't really for me. And I think part of it was also a kind of personal, maybe restlessness isn't the right word, but a curiosity about so many things. And I feel like I have the rare luxury to be able to engage in a bunch of different biological systems in the work that I do. So I can learn about birds and kind of do a deep dive into that world, and then learn about ants, and now learn about phytoplankton in the Arctic. And that's something that I think if you build a career in science, you sometimes get a chance to do crossover to other parts of a system that you study, but you really have to have that motivation to continue studying a fairly narrow aspect of the world we live in for a long time. So I feel lucky that through the kind of work that I do, I get to kind of dive into these places where there are extreme specialists, and then also move between those different arenas.
That's great. Yeah, I think that's a really good summary of the sort of focus that scientists need to take on just to be getting their tenure, and et cetera, et cetera. And I think that it's great how you've kind of been able to allow yourself both a focus, but also, as you said, this kind of curiosity that takes you in so many wonderful directions. And I guess I'd follow up with a question also on the way you make your choices for your projects, because they're always looking at a complex sort of area in biology, in developmental biology, in ecosystems, and how they're all vast and related to so many different factors. So just what prompts you to choose something like this kind of work that you've just done or are working on now about the Arctic, versus like the ant colony, which obviously it seemed like that one kind of fell in your lap, but you might not have wanted to take it up. So how do you kind of get, go from topic to topic?
Yeah, that's a great question. And I feel like I kind of wish I knew the answer a little better. But I think it's some aspect of kind of personal interest, serendipity, and then something that seems meaningful to the moment. So I kind of try to weigh those three things. I think I could be fascinated in creating projects about insects the rest of my life. Like, I would be happy to do that. I just find the world of insects, there's so much to understand and appreciate and admire. So I would be excited to do that. but when another project comes along that has equal interest, I'm also excited about that. I think a lot of it is also a human element. So just meeting other collaborators that at the right time and place that have an interest in bringing a project to life. And that's something that you just can't kind of create a formula for. You have to find people that seem to be on a similar wavelength, that there's a mutual respect for what you can bring to the table. And those things happen serendipitously. I think at the same time, I could imagine doing projects about insects my whole life, but I also tried to think about things that might have some timeliness, whether that's a kind of universal ingredient of reflecting on human communication that now in the course of the colony was made before the pandemic, but since the pandemic it has a whole new layer of meaning about how we connect with other people, and that seems to have a resonance for this time and place. And I think with the project about the Arctic, that's really thinking, what can I bring to the table as a person that is not, redefining policy about climate change, but hoping to shed light on something that is, an important part of our ecosystem and something that needs attention. So I think it's a kind of balance of those three things.
Thank you. That's such a great answer. I really appreciate that. We have a couple questions in the chat. Dina, do you want to speak up and ask your questions, or should I? Vina is one of the leads at the Biodesign Challenge. And if you all don't know about that project, please look up bdc.org, biodesignchallenge.org.
Yeah,
Vina, go for it.
I'm here.
Hi.
I just I can't do video. I hope that's okay.
Totally good.
But yeah, I may have missed this at the beginning, Anna, but I just want to know more about your process of finding your collaborators. Are these people you've worked with in the past? Were you doing like cold calls to say, I'd like you to be my singer, I'd like you to be my musician? Just what was that process like about with reaching out to these people and convincing them to kind of take on this endeavor with you?
That's a great question. And I feel like with all of the collaborators, it's kind of, there's elements of serendipity. So the musicians in particular, Lucy Fitzgibbon, who's the soprano, is somebody that I met in college and has been kind of an inspiration for many of my projects. So a lot of things I write specifically for her because she is just a phenomenal performer. So I feel lucky to know her and that she has the enthusiasm to be a part of these kinds of works. And she was somebody that I lived down the hall from me in college, and then we've been making work together since one of the first projects we made was a project called Bird Brain that was about brood parasites and other bowerbirds. And then she was also in Theory of Flight, and The Colony was created for her. And the pianist, Ryan McAvee McCullough, is her husband and also a collaborator. So I had always wanted to be able to make work for both of them. And then it seems kind of fitting to have them in the context of a performance where they could actually play a couple, perform as a couple, and both theatrically and musically. In terms of the scientific collaborators, I think that has a whole mix of approaches. Sometimes it's people reaching out to me and they might have a project concept. I think in the case of the colony and the affiliated projects that were made from inspiration from the Army Ant Guest collection, that really started with me reaching out to the manager of the biodiversity research collections and at our university, as when I was newly arrived at University of Connecticut, and they were just super welcoming, and it's like, yes, we would be pleased to give you a tour of our insects tomorrow. And then it just happened that they had this collection donated, and one thing led to another, where there became some opportunities to make something a little bit bigger than any of us had originally imagined. And I think that's similar to a lot of the projects, where it just takes kind of the right personalities to come into contact with each other, and then to have some buy-in.
Thank you. That's great. We have another question from Branda, who's here. Branda Miller. Hello.
Hi. Hi,
Hi,
Anna.
Branda.
What a wonderful presentation, and so inspiring. And I mean, it just brings back, yeah, I mean, how many years ago was it when you graduated from RPI with your MFA?
Yeah, like a decade ago now, more than a decade. Hard to believe.
So I'm really fascinated and, really inspired to see your growth, especially, I mean, as your personal identity growth at the core of this transdisciplinary practice, right? Like your media is getting more expanded, your operatic, you always pull these things together like you've been doing this same art in a way for a long time, right? So my
Yes.
question to you is, which is very exciting, is how do you feel that your own representation of your own identity, which is central to these operatic, multimedia experiences, how has your own personal representation evolved and grown in your work as you see your artistic development? And how do you hope to even expand it further as you move forward in the future with yourself centered, in the storytelling experience?
That's a great question, and a complicated question. And I think
Mom's here. My mom likes the question too. Just wanted to share. My mom really loved your work.
thanks for watching. So I guess there's kind of two layers to that question. One is, I guess, kind of, if I'm hearing correctly, kind of like being a performer in one's own work and how that changes. And
Yes.
I think it's something that I reflect on a lot. I think the characters that I play, they have ingredients of me, but also ingredients of other people that I've interacted with, and then they're also just their own people. So I think I'm excited that I just had a child in January, but this is a show about I play a person that doesn't want kids and doesn't have kids, and is kind of cast in this role of a worker ant is kind of how she sees herself, and has mixed feelings about that. And so that's really interesting to see kind of other people especially react to, oh, I thought that was kind of you. Like, I thought Mona was you. I'm surprised that you would want a child. So that's kind of interesting to see. In my mind, the characters that I play are very separate from me, but I hope that they come across effectively in that people think that they're a real life person that has certain hopes and dreams. And I think those things shift through time. So some of the first characters I played, it was more about personal ambition, and then thinking about this kind of social hierarchy. Maybe these are without me having full awareness, like the preoccupations of certain moments in my life. At the same time, I think I have a tendency to kind of mix up work in which I'm at the center and work where I'm not at the center. So kind of alternating a performance with something like an animation where it's like all about the beetles and the lizard. And I'm not a player in that. And I think that's also helpful to me to maybe advance my skills in particular media, and just to have different approaches to thinking about biological systems. So I'm not sure what kind of character I will play next, but for now I'm kind of in a phase where I'm thinking about something very external to me. So thinking about phytoplankton and humanity in the widest sense, where like my personal role is just one of many grains of sand, I guess.
That's interesting how you ended that, because that's how I saw your personality beyond any characters. Somehow you being there is a portal for all of us to be there. It's like a universal connection for all of us as viewers to experience with your characters.
Well, that means a lot that you say that, because that is definitely the hope, is that there's some resonance with the viewer and the audience. Thank you, Branda.
We have another, thank you, Branda. We have another question too from Barbara Latanzi. Barbara, would you like to unmute and, oh, you would like me to read it? Okay, I'm gonna read this. I don't know if you know Barbara Latanzi's work, but she also does amazing animations and other projects. You should know each other. Who teaches at Alfred. Anyway, she says, thank you so much, Anna. I'm sorry that I arrived late. Maybe you have already addressed this question, but the colony integrates insect processes algorithmically and very creatively into the performance. But the processes can be abstracted, as you demonstrated with the MATLAB modeling. Can one describe your Evo Devo approach as a process of hybridization? In other words, is it fair to characterize the approach as a mashup of human behavior itself and insect behavior? How do you study human behavior with this in mind?
Yeah, that's a dense question. I guess my short answer would be yes. Hybridization, I think, makes sense. I think definitely for me, I think the hardest part of creating the work is always this kind of wavering between wanting to be very faithful to perhaps as one example, a generative methodology that is inspired by a biological system, and wanting to be very faithful to the way that that's mapped to the biological system, but at the same time wanting to have results that have an aesthetic experience that I like and hopefully others like, as well as an emotional experience. And I think those things are often in tension. One anecdote that I often like to invoke is, when Stravinsky ventured into 12-tone music, many composers were wedded to an algorithmic process or a generative process where you would go through each of the tones of your tone row, and that was the music that was created. But I think Stravinsky talked about, like, if there was a note created that he didn't like, he would change it, so that he used the generative process as a way to begin the work, at least when he ventured into that. But then if there was a result that he thought could be better in some other way, he was open to making shifts. And I think that's kind of the same. I'm at a place in my work where that's kind of my approach. I use the generative systems or the processes to expand creative possibilities, and also to connect in a kind of meta way to biological systems. But ultimately, I also want there to be a satisfying musical experience and a satisfying emotional arc to the work. And so if things need to be changed in order for that to happen, that's okay. And I think one example of that is that last excerpt I played, where the generative elements are these swells that come and go, so they make appearances. So it's very much this kind of hybrid approach that you describe. I hope that answered the question. How do you study human behavior with this in mind? Hopefully I kind of indirectly answered that. I think kind of the script writing process has that at its forefront. I'm by no means like a cognitive scientist or anything, but I'm thinking about human behavior and reflecting on it in bringing these different ingredients together.
Thank you. That was a great response, and Barbara said so too. So thanks for that answer, and thanks for that question. Any other questions from anyone before we have to sadly tune out here? This has been really an incredible talk, and as I said earlier, but I really appreciate also the way you're discussing the iterative qualities of how the research affects the work and how your collaborations affect the work, et cetera. And how the work affects your work, how it migrates from one kind of project to the next. So you've been so generous with us, and both sharing the work and also in the ways you discuss it. So thank you so much for joining us. We really, everybody just take off your mute and, you know, give Anna a giant thank you.
Art's also appreciated. Thank you so much, Kathy, and thank you everyone for tuning in.
Yeah, and next week is our last Next week is our last BioArt talk with Mary Magic, who's also very magical. So please come for that next week, same time, same place. And thank you all for coming. And thank you again, Anna. So great. So great.
Thanks, everybody.